Okay, so this session about encouraging people to get creative is getting right into the depths of what we're calling the mossy forests, because really out there in pavements and on the moors, the mosses are a wilderness of their own, where they've been a wilderness without direct human intervention. There's lots of discussion about that, but they've been in existence on our planet for millions of years, and the creatures that rely on them and live in them don't really come across the human. The concept of a human being around a moss and the creatures that live in mosses is not really, I don't think, is a thing. It's beyond their imagination, if you like, because the scale of the mossy forest to them is so huge compared to their size. So we thought we'd spend half an hour or so looking at that and thinking about that scale and getting to know a bit more about the creatures that live within the moss. And to start off, Kat's just gathered a few of the drawings that children have done of mosses at that large scale to sort of, and one or two photographs to get us to think about that a bit more. Do you want to show people?
I will, and my photos run into some of the electron microscope images, Naomi. So would you like me to show them as well at the same time?
Yeah, why not?
Thanks. So let me just share my screen. Here we go. Hopefully you can see my presentation. There we are.
Yeah.
So I've just started with a couple of nice photos of moss, really, just to remind us what we're looking at. This one I found in a wood in Cornwall. I didn't cut the heart, but I found it, chanced upon it, and really liked it. Some examples of a moss forest.
Yeah, so this one, you can imagine an ant, say, crawling around that, and it would be a completely different world. And an ant is a creature, of course, that we can see is a few millimeters long, but in that mossy forest, its world would be like a giant prehistoric forest almost. Let's see other photos that things that you've got on there.
So now I've got some photos that some children have done. So these all come from a primary school called Ford Primary in Plymouth, which Jenny and Naomi visited a little while ago. So some of their drawings and sketches of moss and the shapes and some of the creatures they think are in it there as well.
Do you think that one with a person on it, do you think that creature is a tardigrade?
Well, I thought there's it's definite tardigrade looking creature there.
I know we found woodlice. We definitely found a few tiny woodlice that day. And to start off with, a lot of the children were incredibly hesitant to touch the moss because it was dirty. And, there might be soil and there might be bugs and there might be, and then by the end of it, if you remember, Naomi, they were just loving the moss. And so they made little terrariums to take home, really connected with it over a full day. And grew to love it. And we went for a bit of a walk and looked for moss as well in their quite urban neighborhood, and we found a fair amount.
Okay, we did.
And then I've got a couple of photos of the charcoal drawings that have been shared on that Naomi shared on the Bury the Giant Club, which these came from a school that you worked with, possibly Sand School, where you were working with the children there.
No, these came from Morton Hampstead on the edge of the moor, and those youngsters used to passing by mosses on their daily walk to and from school, but they hadn't really thought about sphagnum before and its role in making peat and holding surface, if you like, the surface of the wetland and absorbing all that water. So I got them looking at with a magnifier and being as bold as they could and imagining being very small. So this, I was really pleased with their work, you know, their attention to getting their drawings more and more closely observant, if you like, of the actual plant. Yeah.
And I've got one more, which I think is our personal favourite, because it features the blob moss,
Yeah.
which we all enjoyed this picture. I think it's very sweet.
Yeah, and blob mosses, so if they're the sorts of moss that, I think that was a sort of moss that grows on a roof or grows in a pavement. I've forgotten its name. Don't know whether anybody wants to shout out a common moss that does that, but my books are downstairs ready for tomorrow's session. But anyway, these blobby mosses are quite slow growing if the conditions are sometimes tough. So for instance, on the moor, you can get sort of mounds of mosses that might be tens and tens of years old. You know, they're growing a bit like sphagnum grows a millimetre a year in terms of the peat. If you get a big mound of moss with soil underneath holding it all together, as it were, I think it can be really quite old as a mound. But yeah, we did like this. Do you remember this one, Jenny? Oh no, you weren't there when we did this one, but you've seen it. Yeah, very good.
And then my next photos, they all come from, so we were, Art and Energy were lucky enough to receive some funding to allow us to work with the Electron Microscope Centre in Plymouth, which is part of the Plymouth University. So my next photos all come from that project, where we submitted 6 samples of moss from different locations, some sphagnum, some from a drain, and we were able to get them preserved by the lab. And then we went in and took some photos of them. So these are some extreme close-ups of moss. And then we finished with some possible little creatures that were within those samples, although that part of the project wasn't so successful, and that hopefully if we can do it again, we'll be looking more at trying to preserve some of the creatures and take their photos. But this gives you some of the shapes of the mosses and the structures and the patterns, looking at it, I think, from maybe an artistic point of view, and then trying to zoom in on some of the detail as well and some of the different parts of the structures of the moss.
When we took in the moss to have the samples preserved, I remember Glenn taking the tiniest amounts of moss from these 6, jam jar samples that we'd taken in, and he took the tiniest bit, and I thought, well, they're not going to get much out of that, are we? We're going to get a few photos. And then you start going closer and closer and closer into that surface. And it is like a whole nother planet, a whole nother world. And kind of, I guess, as whether as you become more experienced with this, you become more skillful. But I felt like we were quite just dive in and try, and quite lucky. Well, lots of beautiful things to look at, but it just made you realize, a bit like if you were, I don't know, looking at Earth as you could so easily miss mankind. I suppose not necessarily the effect that we cause, but the chances of actually finding a patch that's got humans on it in a similar way to looking for tardigrades.
Yeah, look at these blobs. I didn't really know what they were. I don't know whether they're maybe Ellis will be able to tell us. I don't know.
I'll keep going. So this is where we start to get I don't know if you can see my cursor moving, but we had a sort of question mark of something here, which we zoomed in on. It's pretty hard to be an expert using an electron microscope and all the different zooms and focus and things. So we've got a couple with possible creatures. So you can see in this one, there's this I think was our best one that Naomi and I found. There's this shape here, which we zoomed in on, a bit fuzzy, but you can see we felt this was potentially a creature. And then last one, some shapes and forms like this, which again we thought were possibly, actually the previous one's better. So we've got lots of these photos, which we have shared on the website, and I will share the link again so you can see them if you're interested. They all have a scale, so you can kind of get an idea of the magnification. And we know which site each moss came from and whether it was sphagnum or another species. That's all of them, I think. Then I'll stop sharing now.
No, just what we're doing is getting people into the mood, thinking about mossy forests, and then welcoming Ellis to take it one step further and talk about the tardigrades, the little mossy pigs, the creatures that do live in the water on mosses and do feature in our mossy carpet project and the stories that we like to tell. So Ellis, can we now hear from you and hear more about the tardigrades and the project and the PhD you're taking, or just explain? It'd be lovely.
Of course, yeah. I'll just share my screen and then full size that. Okay, can everyone see this? Yeah. Okay. Yes. So I'm Ellis. I'm a final year PhD student at the University of Plymouth, and I study tardigrades. So what I'm going to be talking to you about today is sort of raising the question, will tardigrades rule the world? And we'll be going into some insights into these really tough creatures. So what exactly are tardigrades? They're basically microscopic invertebrates, so organisms without a backbone that were first discovered in the late 18th century by a German scientist, Johann Goetze, and he described them as kleine Wasserbar, which roughly translates to tiny water bear, because that's what he thought that they looked like. And there's about 1,300 different species, and you can find them anywhere from the sediments of the deep sea to the meltwater ponds in Antarctica and the moss in your back garden. Now, what makes them particularly interesting to people like me, and I guess most people in the media, is that some species have been shown to survive incredibly extreme conditions. Now, for those who've never really seen a live tardigrade or what they look like, I took a video of one and matched it to some Pink Floyd because it just made it even better. And you can see the tardigrade here. Obviously, it's very zoomed in, but this species that I work on is about 0.1 of a millimeter in length. So if you were to go out, you wouldn't really be able to see these things without a microscope. That's just a little video of what these tardigrades are like. That little green pellet inside, you could probably work out that that's the algae that the tardigrade eats. That's the food that I give it. Probably one of the few people, if the only person in the UK to say I essentially have pet tardigrades that I have to look after on a weekly basis. And so here we have some other tardigrade species just to show you that they come in all different shapes and sizes. So on the top left, this is Ramazotius, which is another moss-dwelling species of tardigrade, or freshwater dwelling, really. And that's one of the more resilient species of tardigrades that we as a group of researchers have looked at. In the bottom right, Patillipes here. This is a marine species of tardigrade, which very little is known about compared to the terrestrial land species. But in terms of differences, you can see these suction-like cups on the end of the appendages, these little suction toes that they have. And we think that the reason that they've evolved those is that it allows them to adhere to the sand grains in the sediment. And that's part of the reason why it can be quite hard to study these marine species, is actually finding them is really, really difficult. And then the bottom left here is the newest species of tardigrade that was discovered, I think, a year, maybe 2 years ago. And those of you who are familiar with the Harry Potter novels will recognize the species name Nagini. So it was named after Voldemort's pet snake, and that's because it has these reduced appendages, so its arms are much shorter, because it's from a sand dune in Finland. And what you find is most organisms that are from sand dunes typically have this reduced appendage feature, because it allows them to move better in that environment. So this is the species that I work on, Hypsibius exemplaris. It's obviously a wild species, but it's been developed as a lab strain now since the late 80s. It actually originates from a pond in Bolton, of all places. Some bloke just went and collected them and then sold them to labs around the world, until sadly COVID essentially destroyed his business. And it's what we call a model organism. What I mean by that is it's quite easy to study the basic biology of this tardigrade. So you can see the adult on the left here. At the bottom, we have the head with the 2 eye spots. You have the mouth parts. You have the storage cells, these little globules that sort of coat the outer side of the tardigrade. So you can see all these structures relatively easily. And on the right, these are the embryos of this tardigrade. So you can see, I don't know if you can see my cursor, but in the center of the screen, this is a 2-cell stage embryo of this organism that I can look down the microscope and study. And within each of these cells is a little dark area. That's the nucleus where the DNA is stored. And you can even see bits of the DNA that's in that nucleus if you've got good eyesight, little dark sort of spots within that circle. That's the condensed DNA. So this is sort of why it's a model. Other tardigrades can be quite hard to see because they're dark in color, their embryos aren't translucent. And, you know, development for them to go from 1 cell to a baby tardigrade is about 4 days. So the turnover time of the generation of these tardigrades is quite fast, which makes them attractive for researchers. I've got a little video here I took of some baby tardigrades that are just about to hatch, moving around inside their shell. So this was taken over a few hours, and I've just sort of sped it up, and you can see the head of this tardigrade here. It's sort of on its front, and this one here. So that's about 4 days in, and it will use its mouthpieces to burst through the shell and come out. Now, essentially what the tardigrades will reproduce in a number of different ways depending on the species. But this one that I work on is asexual, so they're all female, and their offspring are essentially clones. So there's no sexual reproduction, and like a snake sheds its skin, the tardigrade will shed its outer cuticle periodically, and it will deposit between 1 and 10 embryos in that cuticle where they will go on and develop. But the thing that most people are interested in, that I sort of mentioned at the start, was that some species have been able to show this resilience to environmental change or extreme conditions. And the way they do that is by going into a state of dormancy called cryptobiosis. And if we think about a tardigrade on a piece of moss, in a little water droplet, a dewdrop, but it's a hot day and that water begins to evaporate. Now the tardigrade will sense this change in its environment, and it will respond by retracting its head and its appendages and form what's called a tun, which I think is German for barrel. It will lose about 99% of its body water, and its metabolism will entirely shut down. And it's in this tun phase that they can withstand extreme conditions, and they can stay like that for decades at a time. And even their embryos, what you can see in the bottom left here, this is a dehydrated one in box A, are capable of doing this at the 1-cell stage. So they can be stressed as well, just as much as the adults can be. Now, the sort of stressful conditions that have been looked at range quite greatly. So they've been frozen, they've been blasted with radiation, they've been shot into space and exposed to the vacuum of space, and they've even been shot out of a gun, which is what this study in the top left did, where they wanted to test the theory of panspermia, which is where life on Earth started from organisms coming from elsewhere in space onto the Earth. So they wanted to see, could those organisms withstand the sort of velocity of entering the Earth's atmosphere? And that's what they were trying to replicate by shooting these animals out of a gun. And it got lots of media attention, which didn't harm the paper. Now, I'm just going to go off-piste for one slide here, just to mention that while tardigrades get a lot of this media attention, there are other organisms that live within moss, within freshwater environments, that can also withstand these extremes. And one of them is this little fella here, which is called a Deloid rotifer. Now, this Deloid rotifer in particular was discovered a couple of years ago by scientists working in Siberia. They took a sediment core in the permafrost, and when they dated how old that permafrost was, it was 20,000 years old. They took the sample back to the lab and thawed it out, and the rotifer came back to life. So this rotifer has been frozen in time for 20,000 years. That's the entirety of recorded human history that this animal has skipped. Older than the ancient Egyptians, older than, 20,000 years, a long time. And it remained in this dormant phase, only for it to come back to life once we discovered it. Now, these rotifers are known as ancient asexuals, and that's because typically organisms that are asexual don't have the genetic diversity that sexually reproducing organisms do. So their genes aren't as mixed up. And because of that, they often go extinct at a much faster rate, because if there's a change in the environment, they don't necessarily have that diversity in their DNA to allow them to cope. But when we look at the genes and the DNA of these rotifers, they have a lot of genetic diversity, which doesn't make a lot of sense. But what we think happens is when the rotifer enters stressful conditions, maybe it dries out, its DNA breaks up into tiny pieces. Now, usually that would kill most things. What we think that the rotifer does is when normal conditions come back again, they will take bits of DNA from the environment, from its gut, from things it's eaten, and it will sew those bits of DNA from other organisms into its own DNA and sew everything back together again and repair. And that's how it gets its genetic diversity. So this is just an example of, it's not just tardigrades. Although they get a lot of the media attention, there are things like these Deloids which are just as remarkable and worth looking at. Now, what I'm particularly interested in is understanding what are the genes that these tardigrades have that other organisms don't, that allow them to withstand these stressful conditions. And some work has been done on this. One of the more famous papers is this one that came out a few years ago. And what they did was they discovered a gene called DSUP, which just stands for damage suppressor. And we've only discovered it in tardigrades. No other organism on the planet seems to have it, as we know of. And they took a petri dish and put some human cells in one, just normal human cells. And they took a second petri dish with human cells in, but they gave these cells this DSUP gene, this tardigrade gene. They then blasted them both with high levels of radiation, and they found that the human cells that had been given this tardigrade gene were able to replicate and continue as normal. But the human cells that hadn't been given this gene died and weren't able to replicate. So that's one example of ways that we can look at how these genes work. What are they giving to the tardigrades in terms of protection? And therefore, what are the actual applications of this knowledge? So, for example, with vaccines, we all have experienced this recently with COVID, where we go to get your vaccine and they're stored in these fancy freezers that keep them at the perfect temperature. But of course, some of the areas of the world that are developing don't have access to these freezers to store these vaccines. But we know that tardigrades have the ability of drying out almost completely and then coming back as normal again. So can we incorporate genes that allow tardigrades to do that into these vaccines so that they can be dried out and stored and then used in developing countries? Same goes for long-term food storage. And then something that I'm personally interested in is what tardigrades can tell us about neurodegenerative diseases such as Alzheimer's, Parkinson's disease. And that's because in all of our cells, in all animal cells, are tiny organelles called mitochondria. Now, these mitochondria are super important parts of the cell because they provide you with the energy that you need to survive and to perform all the normal processes in your body. Now, when those mitochondria stop working or they malfunction, it can cause things like Alzheimer's, dementia, Parkinson's disease. Now, these mitochondria are also the hub of stress. So when we're under stress, or whether another organism's under stressful conditions or harsh conditions, it's the mitochondria that bear the brunt of most of that. So I want to know what's going on in the tardigrade mitochondria that allows them to withstand the stress. And then can we use that information to help learn about ways that we can, I'm not going to say find a cure, but prolong the life maybe of people that suffer from these diseases. And lastly, in the context of this group and thinking about climate change, what can we do to help conserve our environment using these tardigrade genes? And a good example of this is looking at coral reefs. So we've all seen these horrible pictures of corals that are white, they've been bleached. And that's because coral get their color from an algae called zooxanthellae, which you can see in the top left here. And it shares a symbiotic relationship with this algae. And the algae gives the coral nutrients that the coral needs to survive. And in return, the algae has a nice, safe place to live, away from grazers and nice and close to the surface of the ocean, so it can get light for photosynthesis. However, corals can only live in a small thermal range. So when the ocean's warm, what happens is the algae get stressed and they release toxins into the coral. And this makes the coral get rid of the algae to get it out of them because they don't want those toxins. Now, there's a lot of coral reef restoration projects going around the world at the moment where they're trying to breed more resilient corals, and then they take them out on these sort of plastic piping back into the ocean to help grow the corals again, essentially. But if we can give maybe the algae that suffer from the stress some of these tardigrade genes that would help them cope with higher temperatures in the water, then perhaps we could see less bleaching events and less loss of biodiversity in the future. So I think that's that.
Thank you.
Thank you very much.
Very good.
And if you've got any questions, feel free to ask. I'm happy to answer anything.
Has anyone got any questions? Or put them in the chat?
Yes, I have. It's Alan here. I have a very
Hello, Alan.
Richard Dawkins book,
I'm
The
just going to
Ancestor's
turn my
Tale,
light.
and he mentions Deloitte today, in what I'm reading today. And what a coincidence. And about their reduction strategy, I was trying the female line, and he gives it as mutations that cause the variations that exist, but you've given it a fresh slant. But one of the things that's going through my mind, and you had one of your photographs showed space, men in space. And is it possible that for the long journey to Mars, which some people are going to undertake in due course, no doubt, the radiation damage will be cumulative? Will they be able to use something, that some of the technique that tardigrade has to protect itself, for the spacemen to protect themselves? Is that one of your potential areas of interest or research?
I think that is an area that a section of the community are looking at in terms of partnering with the European Space Agency and trying to understand. I think it's less on the radiation side, because I think that, you know, the actual engineering of the support systems will probably be better attuned to dealing with that issue. I think it's more actually long-term space flight. So obviously these tardigrades can lay in this dormant phase, like cryo sleep, essentially, like you see in the movies, for long periods of time. So I think, I mean, it's this sort of research, particularly with the space stuff, you know, I'll probably be gone before any of this sort of stuff comes to fruition, because it's so complicated and there's a lot of work that needs to be done before that, and obviously ethical arguments that get put in place. So there's a lot to unpack, but I think the majority of the research that's going to be focused on the space side of things will be more long-term, how to deal with long-term space flight.
Thank you for that.
We have a question from Gareth here, which says, when were tardigrades discovered, being so tiny, and how long have they been researched? So I think you said, did you say at the beginning 18th century?
Yeah, so the late 1700s they were discovered by a German scientist, and they've been researched ever since, really. Obviously a lot of the research that was done then, a lot of it's hard to read because it's in different languages, but a lot of it's obviously changed since then. But yeah, I mean, I think a lot of, you know, a lot of study of microorganisms really date from the 1700s because of the invention of the microscope. You know, most of it dates from then on. There was a huge sort of explosion in research and into tiny invertebrates from that point. So yeah, late 1700s.
Brilliant, thank you. And there's a question from Jenny, although we're going to gradually tie ourselves and finish, aren't we, because of the time, but we have got an interest in how did you decide to specialize in tardigrades? I mean, we were amazed by what you've just described, but how did you get into it?
Well, yeah, so I mean, I wanted to be a marine biologist since I was a little boy, and I struggled a bit in school, and the school didn't think I was smart enough to be a scientist and all this, so I had to sort of study from my A levels from home, do it myself. And so, you know, a lot of it was sort of out of spite of my old teachers thinking I couldn't do it. And I got to school and I became obsessed with just the deep sea and extreme environments in general, and how do these things survive this pressure. And then, you know, I went through university and interests change, and I learned about tardigrades, and you know, in terms of animal adaptations to the extremes, they're top of the chain, really. So, of course, you know, that's what I'd want to research. And then this PhD at Plymouth popped up at the right time, and you know, so I went with it. You don't always have much choice with your PhDs, but this one was very broad and allowed me to sort of run with it. So that's how I sort of went down that path.
Brilliant. Thanks. Well, thanks, Ellis. That's opened our eyes to that completely different world. And I really hadn't thought about the fact that there were how many species? 13,000 species?
1,300.
1,300 species. And it's on that note, really, the variety that you get. So you get this massive variety of mosses, but other environments in which tardigrades have lived for thousands of years. You get that sustainability and ability to come back to life of rotifers, but I'm sure tardigrades as well. But it's given me inspiration that all our tardigrades that we're going to make as part of this Mossy, we don't have to make them all the same, do we, Chloe?
Oh, absolutely not. No, yeah.
Oh, they can have little legs and big legs and all sorts. But Chloe, do you want to describe where we're up to with our tardigrades and Jenny as well? But you know, Chloe, do you want to sort of say a little bit about what you've been doing with the tardigrades?
I do, thank you. Yes, so we've been experimenting with making tardigrades. You may have all seen our tardigrade telephone, which is a device that we use for recording messages for the mossy carpet that will be stitched into the mossy carpet. And so we experimented with a range of designs and have sort of settled on this one for the moment, but as you say, we're probably going to deviate. It's a really simple design, actually, although it looks more complicated than it is. It's actually only made with two, this is a slightly bigger piece of paper, two pieces of pattern. So there's a kind of a blob with legs, and then I don't know whether you can see this, there's a second element like that, which makes the underneath of it. So you cut out two pieces of material like this, and then two pieces of material like this. You stitch them together. So there's a join in the middle there, and then a join at the back there, and a join along the legs. And then what you land up with is kind of that sort of shape. And then, so that's stuffed in the middle there. And then we've been stitching around the edges in order to kind of tighten the middle bits and give it that kind of tiry, bulbous, moving kind of feel to it. And so that was the kind of first design, little snouts, little 8 legs that we worked on. And I think Jenny's kind of simplified this a bit further. Jenny, you've been making a whole load of them too, haven't you?
Yeah, so it's evolving. My mum came over the other day and said, why are you cutting out 2 pieces for the kind of belly with that seam up the middle, when actually you don't need a seam up the middle, and then you can stuff it through its snout. So I've been making some using fabric from the scrap store. I'm really enjoying playing. And this one here has grown some moss. Can't get it carried away with embroidering some moss over the top of that one.
Totally darning everything that you can find
I'm
with
hooked.
moss.
And I'm quite enjoying doing a different colour belly to the top sides. Yeah, I'm oh, this one here hasn't been stitched yet, but I really liked this sort of floral when we were looking at those photos earlier on, looking closer up at mosses. Yeah,
that
It's
really excited
very
me,
satisfying.
Jenny, that when you started going two-tone and multicolor,
Hmm.
I found myself thinking, ooh, how do we connect it more with the mossy carpet? And I don't know whether everyone's made a pom-pom for the mossy carpet, but the mossy carpet is covered in fabric, well, covered in pom-poms. And here is, I took a photograph of some of the pom-poms and then I uploaded it onto Spoonflower, which is a digital printing website, and printed it on this eco canvas. And my next tardigrade is going to be made out of something that has a pattern like yours do, Jenny, but has the pattern of the mossy carpet, and when it's finished.
Alan's just commented about draft excluders, and somebody at Scrap Store today was saying about weighing them down so that they could be door stops or draft excluders or something like that as well.
Yeah, my boys and my husband have been asking me to do the same thing. And I kind of feel like you could have tardigrade slippers too, why not?
Yeah, I've done a sort of cyanotype one using some old fabric of that I had that has some cyanotype images of moths from the last project that we did, and mine has got a particularly big snout, but it's also got yellow loopy claws. And now I feel like after Ellis's presentation, maybe we should make a variety of claws, including some sticky. I suppose it's not totally appropriate for mosses, is it? Because those little tardigrades lived in the soil, but
maybe we could put Velcro on the bottom. We've been thinking about how we're going to display all of these tardigrades in November at the Theatre Royal in Plymouth, and we're going to have a big exhibition of how the mossy carpet has got to at the moment, and how we and all of these tardigrades that we're all making. Maybe we need to put Velcro on their paws and stick them onto
we could all move along
a display board. Take them on and off.
Maybe we might end up there. So we have run over rather, because we've been chatting as well as hearing this amazing information about the tiny creatures that live in a mossy forest and beyond, and how resilient they are. They are telling messages themselves to us humans. They're telling us stories about the way we live and the way they live, and some ancient history. I don't know, do you, Chloe, or do you, Jenny, want to talk about the mossy carpet and where we're up to and what we're inviting people to do in the next?
Well, I'm going to talk about very, very briefly the recording your message. So Naomi mentioned the tardigrade telephone, and I don't have a tardigrade telephone at the moment, though the tech is working in here, touch wood. I've begun to add some moss onto some textile moss onto the side of this antique telephone. I hope nobody's too precious. I've been drilling holes to attach it to, and then it's not plugged in at the moment, but when you pick the handset up, you can hear the tardigrade speak, or the tardigrade speak, and it asks you to leave your message for what the tiny things are, moss-like actions you're taking for a brighter, greener future. Then it beeps, you leave your message and pop the phone back down again. So I'm going to be adding a tardigrade onto the top of this one, similar to our other one. And we also have the tardigrade is gone modern, doesn't just have a rotary telephone. The tardigrade also has a mobile number. So I'm going to ask Kat whether she could please pop the number for the tardigrade into the chat. And we would love to invite you to record that message. So my message for this evening, going back to what I was talking about earlier on, might be, I might dial the number, listen to the tardigrade, and then say, hi, I'm Jenny, and I repair my clothes in a very visual way to try and encourage other people to do similar. And that would be lovely as the message, and those will eventually be able to play out of the carpet. So when people interact with the carpet, not only are they inspired by the thousands of people that have joined in with it already, but also over the next few years, but also they will hear voices. They might hear your voice and be inspired, or they might be inspired and think, oh, I already do that, and that's fantastic, and kind of celebrate it. But they might also be inspired and go, I'm not doing that yet, but I could do, and I'm gonna adopt that behavior. So yes, please tell the tardigrade about what you're doing for a brighter, greener future. And I'm going to pass over to Chloe to talk about the textiles element.
So we have this mega ambition for the mossy carpet. We would really like to make 100 meters of the carpet and maybe have 100,000 participants, and maybe it will go to somewhere really, really epic and exciting and celebrate all the things that everyone is doing for a brighter, greener future, and invite everyone to be inspired by mosses in the same way that we have. And so I'm very much hoping that you might be willing to help us by either making pom-poms or textile tufts and sending them to us, or inviting your group or your friends or your family to also have a look at mosses and be inspired to make a pom-pom to add to the carpet. So far, I think I'm brave enough to say that we might have 9 meters already. Do you think that's right, Naomi? Do you think we've got 9 meters of carpet?
Yeah,
so
I would say so.
So over the next 2 months, through October and the rest of September and October, we're going to be running workshops in Plymouth, and anyone who is in Plymouth and able to pop along would be welcome to join us. And details are on our website. Events, all our events are listed on our website. But we're so excited that the whole thing is going to be displayed at the Theatre Royal in Plymouth, because the Theatre Royal gets loads of people through the door. So I really hope that all of those people might also be inspired by mosses and tardigrades and encouraged to come and join us celebrating everything that everyone's doing at this moment in time.
Yeah, thanks, Chloe. So we will wrap up this session about tardigrades, the mossy underworld forests, and the leaving messages, the mossy carpet as well. And hopefully we'll see you all on the 30th of October, when we will be having a multi-subject session about the Bury the Giant and mosses and energy. We'll bring a bit of the science back in, and archaeology as well, the information you can gain from looking deep into peat. So hopefully you'll want to join us for that 30th of October, the same time, from 6:30, aiming to finish at 7:30, but hopefully we will indeed. So thank you very much for coming along, and we will see you in a month's time. Bye-bye.
Thank you.
Thank you.
Thank you. Bye. Nice to see everyone.
Yeah, bye-bye. Bye.